Okay, let us begin our today's topic. Yesterday we were discussing about the structure of proteins. As I told you yesterday, a protein has got different structures. First, the protein forms a primary structure. Then primary structure is followed by secondary structure. Secondary structure is followed by tertiary structure. And then tertiary structure is followed by quaternary structure, right? So this arrangement of protein in primary, secondary, tertiary, and quaternary structure. This is known as the structural hierarchy of proteins, right? So we were yesterday discussing about the primary structure of protein. I told you yesterday what is the primary structure of protein. In primary structure, a protein is in the form of a linear polypeptide chain. This is a linear polypeptide chain. What do you understand by linear? Linear means a chain which is having two open ends, one end and the another end, right? So it is in the form of a linear polypeptide chain. So please don't make noise. One amino acid joins with another amino acid. It joins with another amino acid. So many amino acids join. Many amino acids. Please mute yourself. Put yourself on mute. You are making noise. So many amino acids, they join in a series and they form a long chain of amino acids. This long chain of amino acids, it is known as a linear polypeptide chain. This linear polypeptide chain, it has got a specific number of amino acids in it, and it has got these amino acids in a specific order, right? So in primary structure, a protein is in the form of a linear polypeptide chain having a specific number of amino acids in it in a specific order, right? This linear polypeptide chain, it has got an N terminus and a C terminus. I think you are not knowing about what is N terminus and C terminus. N terminus is the side of polypeptide chain, which is having amino group on it. This is the general formula of amino acid, as you may be remembering. This is one amino acid linked with another amino acid. So in this way, it forms a polypeptide chain. So in this way, amino acids are linked with each other. So it is a chain of amino acids. It is a linear polypeptide chain. One amino acid combined with another amino acid, it is combined with another amino acid, it is combined with another amino acid, and so on, right? So this we call as a linear polypeptide chain. In this linear polypeptide chain in a protein, the number of amino acids for a particular protein is specific. The sequence of amino acids for a particular protein is specific. And this linear polypeptide chain, it has got two ends. One is known as it is N terminus. It is N terminus. Because on this side lies the free amino group. The side of polypeptide chain which has got free amino group is known as N terminus. And this side of polypeptide chain, which at the end has got free carboxyl group, it is known as C terminus. It is known as C terminus, right? So in primary structure, a protein is in the form of a linear polypeptide chain having a specific number of amino acids in a specific order. This linear polypeptide chain, it has got two free ends. On it is one free end, there is free amino group. So this free end of the linear polypeptide chain, while we have got free amino group, we call this side of polypeptide chain as N terminus. And this end of the polypeptide chain, where there is free carboxyl group, this is known as SC terminus. So this is the primary structure of protein. As I told you yesterday, proteins are not usually functional in primary structure. Proteins are not functional in primary structure, generally. However, there is a protein known as insulin protein, which is just formed of two polypeptide chains. Polypeptide chain A, polypeptide chain B, right? In A polypeptide chain, there are 22 amino acids. In B polypeptide chain, there are 31 amino acids in human insulin. So in human insulin, there are total 53 amino acids. And this human insulin, it is formed of two polypeptide chains. And these polypeptide chains, they are linked by disulfide bridges or disulfide bonds, right? So this insulin is the only protein which is functional in primary structure. Otherwise, the remaining proteins are not functional in primary structure. So that is about the primary structure of protein. Now we will move ahead and we will see the secondary structure of protein. In secondary structure, a protein exists in three different forms. Now we will see secondary structure of protein. In secondary structure, a protein exists in three different forms. In secondary structure, a protein may be in the form of an alpha helix. A protein may be in the form of an alpha helix. Or in secondary structure, a protein may be in the form of a pleated sheet. Pleated sheet. Generally, beta pleated sheet is found. In secondary structure, a protein may be in the form of a pleated sheet. Most commonly found is beta pleated sheet. Or a protein in secondary structure may be in the form of a triple helix. So after primary structure, a protein assumes a secondary structure. In secondary structure, a protein may exist in three different forms. It may be either in the form of alpha helix, or it may be in the form of a beta pleated sheet, or the protein may assume the triple helical structure, right? So first we will see what is an alpha helix, how an alpha helix is formed, and which proteins exist in alpha helical form, right? So first we will see what is an alpha helix, how these alpha helical structures are formed, and which proteins are in alpha helical form, right? So let us see alpha helix. As the name indicates, helix. Helix means in the form of a coil. So an alpha helix, it is a long chain of proteins. Sorry, it is a long chain of amino acids. Alpha helix is a long chain of amino acids, which is coiled around an imaginary axis to form a helix-like structure, to form a helical structure. Suppose this is one amino acid. Do you know what is the general formula of amino acids? NH2, C, H, R, and COH. NH2, alpha carbon, carboxyl group. Do you remember general formula of amino acids? NH2, C, H, R, C double bond OH. So amino group, alpha carbon, carboxyl group, hydrogen atom, and R, right? So we here do not consider these R groups and hydrogen atom. In this form, in this structure, we will not consider R groups and H groups. We will only consider carboxyl group and amino group. So this is the carboxyl group, this is amino group, right? So one amino acid, it is linked with another amino acid. It is linked with another amino acid, which is linked to another amino acid. It is linked with another amino acid. Just a minute. One amino acid, another amino acid, is another amino acid, This is another amino acid. Good. Then there is another amino acid. This is another amino acid. Link it to another amino acid. Link it to another amino acid. So in this way, one amino acid, NHCCO, this is one amino acid. It is linked to another amino acid, NHCCO. It is linked to another amino acid, another amino acid, another amino acid, another amino acid. So many amino acids, they are linked to each other and they form a polypeptide chain first. They form a polypeptide chain first. Do you understand what I mean by this NHC and COH group? As I told you, a protein, an amino acid has a formula NHH C H R C double bond OH, right? So this is the general formula of amino acid. In this example, we only consider NH group, we consider carboxyl group, and we consider the central carbon atom known as alpha carbon. So this is the amino group, this is carbon, this is carboxyl group, one amino acid. Amino group carbon carboxyl group, another amino acid. Amino group carbon carboxylic acid, another amino acid. Amino group carbon carboxylic group, another amino acid. So in this way, many amino acids combine and they form a polypeptide chain. They form a polypeptide chain. Now this polypeptide chain, it curls around an imaginary axis. It coils around an imaginary axis and it forms a helical structure. It forms a coil-like structure. This coil-like structure, which it forms like this, right? This becomes the alpha helix. This becomes the alpha helix. So now the question arises, how this helix can be maintained in its helical form? How this helix can be maintained in its helical form? So it is maintained by hydrogen bonds. It is maintained by hydrogen bonds. Hydrogen bonds are formed between the amino group of first amino acid and the carboxyl group of next fourth amino acid. So this is amino group. This is first carboxyl group, second carboxyl group, third carboxyl group, fourth carboxyl. So amino group of first amino acid, it will form hydrogen bond with the next fourth carboxyl group. Amino group forms a hydrogen bond. Forms a hydrogen bond with next fourth carboxylic group. So this is the first amino group. It will form a hydrogen bond with fourth carboxylic group. This is first carboxylic group. This is second carboxylic group. This is third and this is fourth, right? Similarly, this amino group, it will also form a hydrogen bond with next fourth carboxylic group. This is first carboxylic group, second carboxylic group, third and fourth, right? So this amino group will form hydrogen bond with the oxygen of this carboxylic group, right? Similarly, this amino group, it will form a hydrogen bond with next fourth carboxylic group. This amino group will form hydrogen bond with next fourth carboxylic group, right? This amino group will form hydrogen bond with next fourth carboxylic group. So in this way, hydrogen bonds are formed between amino acids in this alpha helix. So when hydrogen bonds are formed, they stabilize this helix. They stabilize this helix. They do not allow this helix to get open. They do not allow this helix to get loose, right? So they stabilize the helix. Do you understand what I am saying? I will change this into a helical form. I will make a helix of this, right? This is the helix I am making of it. I made it into a helix. I made this piece of cloth into a helix, right? It assumes the helical shape. But if I will leave it, it will uncoil. It will uncoil. So it will lose its helical form. So how this linear polypeptide retains its helical form. How this linear polypeptide remains in its helical form, right? So this linear polypeptide, it remains in helical form by the help of hydrogen bonds. So hydrogen bond is formed between first amino group and next fourth carboxyl group. This is first carboxyl group, this is second carboxyl group, this is third carboxyl group, and this is fourth carboxyl. So hydrogen bond will be formed between oxygen and hydrogen of first amino group and the next fourth carboxyl group. Similarly, this amino group, it will form hydrogen bond with the next fourth carboxyl group. First carboxyl group, second carboxyl group, third, fourth. So they will form a hydrogen bond. Similarly, this amino group will form hydrogen bond with next fourth carboxyl group. First, second, third, and fourth, right? This will form with next fourth. First, second, third, and fourth, right? This will form with next fourth. First, second, third, and fourth, right? So in this way, hydrogen bonds, they are formed between the amino acids, between the amino group of first amino acid and the next fourth carboxyl group. So these hydrogen bonds, they maintain the shape of this alpha helix, right? Now, what is an alpha helix? How an alpha helix is formed? So for the formation of alpha helix, first the amino acids, they combine in a linear manner. They form a long polypeptide chain. So many amino acids combine, they form a polypeptide chain. Then this polypeptide chain, it gets coiled around an imaginary axis. It gets coiled around an imaginary axis. So it becomes a right-handed helix. It becomes a right-handed helix. It turns rightwise. So when it assumes a helical shape around an imaginary axis, then what maintains its helical form? How its helical shape is maintained? Its helical shape is maintained by hydrogen bonds formed between the first amino group and the next fourth carboxyl group. The first amino group, it forms a hydrogen bond with the next fourth carboxyl group. First carboxyl group, second carboxyl group, third carboxyl group, fourth carboxyl, right? So in this way, it assumes a helical form. And this helical form, it is known as alpha helix. In this alpha helix, there are approximately 3.6 amino acids. 3.6 amino acids per term. Per term. There are approximately 3.6 amino acids per term. So one amino acid, another amino acid, another amino acid, another amino acid. So there are approximately 3.6 amino acids per term, right? So this becomes the alpha helical structure. Now, which proteins are in alpha helical structure? Can you name any protein which has got alpha helical structure? This alpha helical structure, it is found in keratin protein. Keratin protein. It is found in keratin protein of hair, wool, and nails. We have got keratin protein in our hair, right? We have got keratin protein in the wool of animals. And keratin protein is also found in the nails. So this keratin protein of hair, wool, and nails, it has got this alpha helical structure. This keratin protein of hair, wool, and nails has got alpha helical structure, right? So that is all about alpha helical structure. I hope now you understand what is the alpha helical structure. So in alpha helical structure, first amino acids combine, they form a long linear polypeptide chain. Then that linear polypeptide chain gets coiled around an imaginary axis. It assumes the helical form, it assumes the shape of a coil. Then that coil is stabilized by formation of hydrogen bonds between the first amino group and the next fourth carboxyl group. So hydrogen bond is formed between oxygen and hydrogen of first amino group and the fourth carboxyl group. So in this way, this amino group forms hydrogen bond with fourth carboxyl group. This will also form hydrogen bond with next fourth carboxyl group. This will also form hydrogen bond with next fourth carboxyl group. This will also form hydrogen bond with next fourth carboxyl group. So these hydrogen bonds, they maintain the shape of this alpha helix. And we find this alpha helical structure of protein in the keratin protein of hair, wool, and nails. So that is all about alpha helical structure. So this is the secondary structure. So alpha helix is the secondary structure of protein. Now after this, we will discuss pleated sheet, beta pleated sheet structure. Did you understand this? Any questions? Any doubt about this? Now, in secondary structure, a protein may also be in the form of beta pleated sheet. Beta pleated sheet. This beta pleated sheet is formed when small peptides lie parallel to each other, right? I will show here the example. This is NH, CH, R, C double bond O. This is one amino acid, isn't it? One amino acid? Amino group, alpha carbon, hydrogen atom, R group, and carboxyl group. One amino acid. It gets linked to another amino acid. NH, CH, R, C double bond O. Another amino acid. It gets linked to another amino acid. NH, CH, R, C double bond O. Right? So this is a small peptide chain. There may be another amino acid, so I will draw only this much. Now it gets linked. This is another another peptide chain. C, H, R, NH. C double bond O, C, H, R, NH. C double bond O, C, H, R, NH. One amino acid. Amino group, alpha carbon, hydrogen atom, alkyl group, carboxyl group. Another amino acid. Amino group, alpha carbon, hydrogen atom, alkyl group, carboxyl group. Another amino acid. So similarly, many such amino acids, they come to lie parallel to each other. This is another amino acid. Right? So this is a small peptide, this is another small peptide chain, this is another small peptide chain, right? Now this peptide chain, in this peptide chain, hydrogen and oxygen atoms, they form the bonds. Hydrogen bonds are formed between hydrogen and oxygen. This is a hydrogen bond, this is a hydrogen bond. Similarly here, this is a hydrogen bond, this is a hydrogen bond, right? And there are also hydrophobic interactions between their side chains. This is alkyl group, hydrogen atom. They also form interactions. They also interact with each other. And these are known as hydrophobic interactions, which also hold them together. Right? These are hydrophobic interactions. So in this way, a small peptide, another small peptide, another small peptide chain, right? They come to lie side by side. They come to lie side by side. And they form an extended sheet-like structure. They form an extended sheet-like structure. This sheet-like structure is known as beta-pleated sheet. This is known as beta-pleated sheet. In alpha helix, amino acids combined with one another. They formed first a long polypeptide chain. Then that long polypeptide chain became coiled around an imaginary axis. That led to the formation of alpha helix. But in beta pleated sheet, many small peptide chains, they come parallel to each other. They come parallel to each other. This is one small peptide chain. This is another small peptide chain. This is another small peptide chain. There may be some 3 to 10 amino acids per chain. There may be some 3 to 10 amino acids per chain. How many amino acids are present in this chain? Can you tell me? How many amino acids are present in this chain? NH, alpha carbon, hydrogen atom, alkyl group, carboxyl group. One amino acid, right? This is one amino acid. From here it is another amino acid. From here it is another amino acid, right? So in this example, there are 3 amino acids in one peptide chain. Then similarly, another peptide chain with 3 amino acids. Another peptide chain with 3 amino acids, right? So they come parallel to each other. They get joined by hydrogen bonds formed between hydrogen and oxygen of amino group and carboxyl group. Hydrogen bond is formed between, this is a hydrogen bond. This is a hydrogen bond. This hydrogen bond is formed between amino group and carboxyl group. 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 So these hydrogen bonds, they keep these chains joined to each other. So in this way, many such peptide chains, they come parallel to each other. There may be 3 or 4, right? They come parallel to each other. They get linked by hydrogen bonds and they form an extended sheet-like structure. So this extended sheet-like structure, it is known as a beta-pleated sheet. In this beta-pleated sheet, a single peptide chain may possess anywhere between 3 to 10 amino acids. One peptide chain may be 3 to 10 amino acids long. Another peptide chain may be 3 to 10 amino acids long. This peptide chain may be also 3 to 10 amino acids long. So in this way, they form an extended sheet-like structure. And this sheet-like structure is known as beta-pleated sheet. So which protein exists in this beta-pleated structure? Have you heard about the fibrion protein of silk? Fibrion protein of silk. We have got silk. In that silk, we have got a protein known as fibrion protein. That fibrion protein, which is present in silk, is in this beta-pleated sheet structure. The fibrion protein of silk is having this beta-pleated sheet structure, right? It may be also found in the proteins of feathers of birds. Feathers of birds. The keratin protein, which is present in the feathers of birds, that exists in the form of a beta-pleated sheet structure, right? Now, I hope you have understood what is alpha helix and what is beta-pleated sheet. In alpha helix, first a linear polypeptide chain is formed. Then that linear polypeptide chain gets coiled around an imaginary axis. That becomes helical. That becomes a coil-like structure. Then that is stabilized by hydrogen bonds. So we call that as alpha helix. In beta-pleated sheet structure, small peptide chains measuring some 3 to 10 amino acids in length, right? Small peptide chain of 3 to 10 amino acids long. Such 3-4 peptide chains come parallel to each other and they get linked by hydrogen bonds formed between amino group and carboxyl group, amino group and carboxyl group, right? So in this way, they form as extended sheet-like structure known as beta-pleated sheet structure. So this beta-pleated sheet structure, it is found in fibrion protein of silk. If you will find it difficult in examination to draw this diagram, you can simply represent a beta-pleated sheet by a diagrammatic representation. We can also represent this beta-pleated sheet simply. I will show you how you can represent this beta-pleated sheet in a simple manner. You may find it a bit difficult. It is not difficult. If you understand, then it is not difficult. Draw 3 lines, 3-4 lines. Right? Draw a line this, like this. this point. Let's extend this to this point. So this is the diagrammatic representation of a beta pleated sheet. So this represents one polypeptide, another polypeptide, another polypeptide, another polypeptide, another polypeptide, right? So you can draw 3 or 4 polypeptides in this way. And these 3 or 4 polypeptides, they are linked by hydrogen bonds between their amino groups and carboxylic groups. And this is the diagrammatic. We say it is diagrammatic representation of beta pleated sheet. Beta pleated sheet. Right? This is the simple way to show a beta pleated sheet. This is the diagrammatic representation of a beta pleated sheet. And this beta pleated sheet structure is present in the fibrin protein of cell, right? Similarly, in secondary structure, protein may exist in the form of a triple helix. Should I rub this? Should I rub this?
Yes, sir.
Okay. In secondary structure, a protein may exist in the form of a triple helix. So first, amino acids join. How many amino acids join? Some 10,000 amino acids. Some 10,000 amino acids. They may join and they may form one polypeptide chain. Similarly, another polypeptide chain of some 10,000 amino acids joins with it. Right? So two polypeptide chains join. Then to this, another polypeptide chain may be added. Another polypeptide chain of some 10,000 amino acids may be added to it, may join to it, right? So in this way, three polypeptide chains, each formed of some 10,000, some 1,000 amino acids, sorry. In this way, three polypeptide chains, each formed of some 1,000 amino acids, may join together and they form a triple helical structure. So first one polypeptide chain is formed, right? Then to this another polypeptide chain joins, right? Then to this another polypeptide chain joins. So three polypeptide chains, they get intertwined. Over each other and they form a structure known as triple helix. This structure is known as triple helix. So where we find this triple helical structure? This triple helical structure is found in tropocollagen protein. It is found in tropocollagen molecule or tropocollagen protein. We call this tropocollagen molecule as simply as a collagen molecule. We may also call it simply as a collagen molecule. So a collagen molecule is formed of three helices. First, a linear polypeptide chain formed of some 1,000 amino acids is formed. Then another linear polypeptide chain of 1,000 amino acids. Then another linear polypeptide chain of 1,000 amino acids. They get intertwined over each other. And these three linear polypeptide chains, when they get intertwined over each other, they form a triple helix. So this triple helical structure is found in tropocollagen or collagen molecule. Then many such collagen molecules. This is one helix, this is another helix, this is another helix. So this is one collagen molecule. Now another collagen molecule. One helix, another helix, another helix. Another collagen molecule. One helix, another helix, another helix. Another collagen molecule. One helix, another helix, another helix. When many such collagen molecules combine, they form a collagen fiber. And bundles of these collagen fibers, they are found in our connective tissue. These collagen fibers, they are very much tough. They are very much tough and they are inelastic. And they are inelastic. These collagen fibers, they are very much tough and very much inelastic, right? And I hope you know perhaps this collagen protein, it is the most abundant protein in animals. It is the most abundant protein in animals. This forms 25% to 35% of total protein content in an animal. Collagen protein is the most abundant protein in animals. It forms 25 to 35% of protein content, total protein content of an animal, right? And this collagen protein, it is basically formed of tropocollagen molecules, and each tropocollagen molecule is a triple helix. So amino acids first combine in a linear manner to form a linear polypeptide chain. Then three such linear polypeptide chains, they intertwine over each other. And they form a triple helix. Then many triple helices combine. One triple helix, another triple helix, another triple helix, another triple helix. Many triple helices combine and they form a collagen fiber. Then many collagen fibers combine, they form collagen fiber bundle, which provide toughness to our connective tissues, which provide strength to our connective tissues, right? So they are found in bones. They provide strength to the bones. So these collagen fibers, they are very much tough and inelastic. And this collagen is the most abundant protein found in animals. It forms about approximately 25% to 35% of total protein content of an animal. I think we are running out of time now. It is 11:06. So this is section for today.